First things first, on behalf of the Cambridge Center for International Research, I would like to welcome Professor Ardem Patapoutian to our Student Research Symposium. Professor Ardem Patapoutian is a Lebanese-American molecular biologist, neuroscientist, and Nobel Prize laureate of Armenian descent. Currently, Professor Patapoutian is a neuroscience professor at the Howard Hughes Medical Institute, as well as the Scripps Research Institute in California. In 2021, Professor Patapoutian won the Nobel Prize in Physiology and Medicine jointly with Professor David Julius, and it is our honor and pleasure to welcome him today to speak to our students. First things first, let's get a warm round of applause for.
Thank you. Thanks so much for the invitation. I will talk to you mostly about science. I think organized the talk in a way that most of you high school students should understand it. But I also wanted to start with one slide on my career path, and I know there will be time for questions and answers at the end, and I'm happy to talk about the science, the process of becoming a scientist, or anything you really want to. So as you just heard in the introduction, I'm Armenian origin, and I grew up in Lebanon. So the civil war in Lebanon, started when I was 8 years old, and so pretty tough childhood. Growing up, I didn't even know that science as a career was an option. So I came to United States, to California, as a pre-med student, so going to medical school because that was the only thing I thought was available to me in the sciences. And then something kind of funny happened. I was going to UCLA, this very large college in California. So my classes were full of students, like 300 students. So I felt like I was not getting to know my professors, and I needed letters of recommendations to go to medical school. So I decided, cynically in a way, I guess, to work in a laboratory just simply to get a good letter of recommendations. I had no interest in actually doing science. Taking lab classes at UCLA, I thought I didn't like it actually. But working in a lab kind of completely changed my life. I just realized that I really loved it. Immediately kind of shifted my focus from going to medical school, which of course is a wonderful profession. I'm not putting medical school down. It's just what fits individuals is very different. And so my, best advice to all of you is find a line of work that does not feel like work. I love doing science. I love the process of it. It's true that getting external validation, like prizes, including the Nobel Prize, is wonderful. But if you don't enjoy the process, then working very hard, being miserable for a goal doesn't make sense. So, and my example is I think interesting as a way to make sure that you really explore different options. Take my example as not liking lab classes, but absolutely loving working in a lab. So it would have been so easy for me to say, no, I don't like this, and not even try. The way I got to it is so indirect, fortuitous. And I think about this a lot, and I have a college-age son, and I also think about, how much effort in the United States at least goes into deciding what college to go to instead of finding your passion of what you should be doing. So I encourage you to think about that. So what I'm going to tell you about is work that we do in my lab. And my lab is usually 15, 16 people. They're mainly graduate students, so PhD students and postdoctoral fellows, some undergraduate researchers, even some high school students in the summer. But this was just from a lab reunion a couple of years ago, bringing not just current, but previous lab members. So we had a lot of fun. The other way I like to present people who've come to my lab is on this map. So each blue target is the birthplace of one graduate student or postdoc who came through my lab. And as you can see, Science is a very international affair. They come from all over the world, and I personally love this diversity. We're doing better in the northern hemisphere than southern hemisphere for diversity, but we're working on it. So the science that we do is something very intuitive for you to, I think, understand. How do we sense touch? So if you compare it to other senses, such as taste and smell, are based on chemicals, sensing chemicals. Vision is based on sensing photons. But hearing and touch do something very different. They change this physical stimuli like pressure and temperature, and in some aspects chemicals, into the sensation that we understand as the sense of touch. And these are just particular examples that, again, you take for granted, but we are very particular about our environmental temperature. So much so that most car companies now have designed dual zone temperature control in cars so that the driver and the passenger would enjoy different temperatures. And hopefully you're all too young to enjoy wine, but wine is stored and drank at very different temperatures based on what kind of wine it is. So this just shows how picky we are when it comes to temperature. Touch is absolutely fascinating. Your ability to discriminate mechanical stimuli at your fingertips extends into the nanoscale. So you can sense 1/500th of the diameter of a hair cell indentation by just running your finger over it. But touch is not just measured in this quantitative sensitivity. We all realized this during COVID years that we rely on what we call affective touch for our emotional health. And this related sense of proprioception, which I sometimes call our most important sense, but most of us don't even know that it exists. This is, instead of sensing your skin, you're sensing how much your muscles are stretched. And from that information, you have a very clear image of where your limbs are in space. This is how I can close my eyes and touch my nose. If you think about how did I just do that without seeing, it just this almost unconscious sense of how much your muscle stretch translates in your brain into an image of where your limbs are, and you have very fine control over it. This is how you can simply walk, or as these two musicians demonstrate, they're performing complex tasks like playing an instrument without even looking at their fingers. But to me, the most powerful image for touch is this one. This is a little boy who's visiting President Barack Obama in the White House. And he tells the president that other kids tell him that his hair looks like the president's. And Mr. Obama just bends over and says, "Touch it, dude." And the idea here, of course, is not just that he's sensing the texture, but this powerful image, this affective touch of realizing for this boy that someone who not just looks like him, but his hair feels like his, is the president of the United States. So all of these that we talked about, touch, temperature, and also pain, are all related senses, sometimes called the fifth sense, coming from the days of Socrates. So we didn't talk much about pain, but acute pain, hammer hitting your finger, is essential for survival. So you learn not to do things that are harmful for you. But chronic pain is a major unmet medical need in our society. All you have to do is follow the opioid epidemic in the United States and other places, and represents pain when the stimulus that causes it is no longer there. So it's kind of when pain goes awry. So to study all of this, we need to study the sensory neurons that actually detect these stimuli. And they are a group of what we call peripheral nervous system. So we distinguish your brain, which is a central nervous system, to peripheral nervous system, which kind of looks at your body overall. And this is a very cool video from a zebrafish embryo. Each of these little balls are a group of about 10,000 neurons that we're talking about. And during development, they send these processes all over the body where they're detectors of various physical and temperature stimuli coming from the body and the skin as the environment. These neurons are fascinating. If I just look at one of these neurons, and this is a very tall basketball player, ridiculously holding its own neuron, but this neuron's length, because let's say if it's in the lumbar region, it sends a peripheral extension to the toe, and the other end of neuron has to go all the way to the brain. It's one of the longest cells in your body, very thin, but very, very long. And their job is pretty simple. They, in the skin, at the nerve endings, they're specialized proteins that sense mechanical force or temperature and send this information to the brain. So that's what our lab was interested in. These sensory receptors that translate this information was not known. We knew that they're a specialized protein called an ion channel, but its identity was not known. So you know that we know of our genome, we have 20,000 genes approximately, but we don't know the function of each of them yet. We know some of them, and this one was not known. In case you have not too much biology background, proteins, of course, can be anything. They could be hormones, insulin, keratin, structural proteins in your skin, for example, or there could be sensors like ion channels. So ion channels are fascinating molecules, I think. You can think of them as electrical switches in neurons, and they do something actually very simple. They sit at the plasma membrane, and they are either closed or open. When they're closed, they're doing nothing. When they open, ions flow in, and this just shows the incredible efficiency of it. 10 million ions can go through one ion channel protein in 1 second. So once ions flow into the cell, it changes the electrical property of the cell. We call it the neuron becomes depolarized. And this causes an electric impulse to go through the neuron. And then the message travels from one neuron to another to the brain. So they're kind of, as I said, electric switch of the nervous system. And ion channels have been targeted for a variety of drugs in the pharmaceutical company. In the market, there are drugs that affect ion channels that ameliorate anxiety, CFTR, cystic fibrosis, epilepsy, stroke, etc. So now we've reduced the question of how you sense mechanical force to temperature to asking which ion channels do this job. Again, just to give you a sense of how tiny these molecular machines are, a grain of rice is about 500 times bigger than a cell. And in the cell, I tried to write a tiny dot here where the ion channel would be. You can't even see it, it's that small. Ion channels are 10,000 times smaller than a cell. So submicroscopic elements. So how do you go about finding them? I'll skip this. So we've done this both for temperature sensors, and I'll quickly talk about two of them, TRPM8 and TRPA1, that are involved in cold and burning sensation. And I'll talk about piezo's in touch and pain and other systems. So the way we do this is you use molecular biology to find these ion channels in usually cells, and then go to either clinical studies or in mouse genetics to find out what they do. So one of the ways you can record from ion channels is called patch clamp electrophysiology. It's, again, a remarkable technique that's been with us for ages. I told you that when ion channels open, they cause an electric current in a way. But these currents are tiny compared to the currents that you're used to. Picoamps, which is 10 to the power minus 12 amps instead of single amp, let's say, for a 100-watt light bulb. And with these very sensitive machines, we can actually read out single ion channels. This is one protein that I told you that you can hardly see, opening and closing. So these little red deflections that you see is single ion channels opening when you cool the cell from 30 degrees centigrade to 18 degrees. So at first you can see single channels opening. As it gets colder, you see multiple channels open at the same time. So when we identified TRPM8, for example, we knew that we had the right thing because we can record from it like this. Very interestingly, the cold activated ion channel that we found is also activated by menthol. So menthol is an active compound in mint leaves, and it's described as a cooling, minty, fresh sensation in the mouth. And interestingly, menthol is used in a variety of products that you might use, pain rubs, toothpaste, things that give everything that you use that gives you that cooling sensation, for example, in your mouth while you're brushing your teeth, is because this has a compound, menthol, that activates the same channel that is actuated by cold itself. That's why menthol feels cooling. So this is one of the fun facts of our science, where this compound that has been used for decades for cooling purposes ended up being, we know now why, because it activates the same receptor channel that actually cooling does. In animal models, I'm not going to show you, but tryptamate is required for cold thermosensation. So if you engineer a mouse without tryptamate, they cannot distinguish cool from normal room temperature. So normally mice, for example, would avoid cool temperatures. They don't like it. These mice just can't tell. So another ion channel that we found, initially activated by cold, but now we know it plays more a role in giving you this burning sensation. And similar to menthol, many chemicals, for example, in mouthwashes that cause this very strong burning sensation in the mouth. If you like sushi, that wasabi that you use with it, that gives you that very strong burning in the mouth and the nose, as well as garlic. All of these compounds ended up being activators of TRPA1. So this is all fun and good, but it actually is interesting because we figured out by these experiments and others that TRPA1 is actually sensor of reactive chemicals and is a general sensor for pain. And we cloned or identified TRPA1 in 2002. 20 years later, blockers of TRPA1 are now in clinical trials for various pain indications. Okay, the rest of the time, I just was going to talk to you about mechanosensation. And this is what the Nobel Prize was given for. And the work started by Bertrand Coste. He was a postdoctoral fellow in my lab. This is about 10 years ago, the picture. And now he runs his own lab in Marseille, France. And here I'm going to tell you about how he found the channel, just because there's interesting lessons there. So how do you even assay mechanosensation? Temperature, it's easy, right? You have a cell, you put cold temperature on it. For mechanical stimulation, what we do is assays like this. On the right, you see this recording pipette, which is stuck to the cell and is recording the voltage differences. And on the left, the glass probe that keeps pushing the cell is very carefully, at very quantitative levels, is pushing the cell at 1 micrometer at a time. And if the cell expresses a mechanically activated channel, you can then record currents like the one you see in the lower right. The upper lines show how much you're pushing the cell. The lower lines is actually the current you're recording from the cell by pushing the cell more and more. So how do you go from this assay to finding the channel? Bertrand screened and found a cell that grows in a dish that's mechanosensitive. From using lots of bioinformatics, gene expression, etc., he thought that there are about 300 candidates that could be the channel. And then he took the very difficult process of what we call knocking them down using this technique called RNAi. So you can knock down each gene at a time and see, if I take this gene out, does this mechanically activated currents go away? And this is his data. On the x-axis, you can see the amplitude of the current that he can record. And on the y-axis, it's the results of knocking out one candidate gene at a time and seeing if the current is gone. So to get this kind of error bars, Bertrand had to spend about 2-3 days on each candidate to test them. So if you do the math, for the first 11 months that he worked, this is full-time job. Imagine every day you're doing the same thing. And for 11 months, you get one negative result after another, but you don't give up. And the 72nd candidate that he knocked down was called unknown family 38 member A. In red, you can see when he knocked that down, he saw massive downregulation of the mechanically activated currents. Bertrand and I renamed FAM38A piezo1 from the Greek piezi, meaning pressure. So one of the things that we have done is solve the structure of piezo1, and it's a beautiful, very large molecule. So I told you already that ion channels are in transmembrane domains. That means that they have to have these helices. You can see them, little ribbons that have to cross the membrane up and down so that it lays in the membrane. So most ion channels have 2 to 6 transmembrane domains. Piezo has 38, more than any other protein in your genome. And then on top of it, 3 of these proteins come together to form a trimer. So each is colored differently. So the proteins touch each other at one end, and at the center is where the pore is. And when the channel opens, the ions will go through there. You can see that it has this unusual curved shape. That's actually its mechanism of function. So it sits in the plasma membrane like this. Blue is the plasma membrane. In reddish color is the piezo channels kind of digging a hole and sitting in there. And they're perfectly evolved to sense tension. So that if you stretch on the cell, give it extra tension, this grooves kind of flatten, which causes the activation of the channel. So it's a very mechanical process. So we have done experiments in mice to show that piezo2, one of the piezos that we found, is required for not just for sense of touch, but also for the sense of proprioception, the sense that we talked about that requires you to have coordination and balance. And interestingly, our collaborators at the National Institutes of Health actually identified a small population of individuals who have mutations in piezo2. Interestingly, initially they were all diagnosed as having muscle or motor neuron problem, but when we looked at them carefully and sequenced their genome, they found that they have mutations in piezo2 and they lack touch and proprioception, just like we had shown in the animal models. So this is a video of one of these individuals that doesn't have piezo2, and this is a test of proprioception. She's blindfolded and asked to walk in a straight line. Now, you and I can do this very easily. As you can see, she's harnessed for her own protection because she is incapable of doing this. How about pain? Acute pain, so I mentioned hammer hitting your finger, is normal in both animals and in the individuals without piezo2. But interestingly, we had shown that tactile allodynia, which is when touch becomes painful. So you experience this, for example, after sunburn or injury, that just touching your shoulder, for example, will be painful. This is a nuisance, but for people suffering from neuropathic pain, this is a major medical condition. And we have shown both in animals and in humans, which is shown here, that this tactile allodynia, this specialized kind of pain that's clinically relevant, is completely gone in individuals without piezo2. So in this experiment, again, the individuals are blindfolded and you have this box A and B on the arm. One is placebo, gets nothing. The other side gets capsaicin, chili peppers, and heat. So you get inflammation. And then control individuals, when you touch point A and B, they all get correct that now B is painful. But individuals that don't have piezo2 can't sense this pain at all. And if you ask them, you have to guess A or B, because that's how the assay works, they guess randomly and get it right 50% of the time. Okay, so I told you that piezo2 is involved in somatosensation, and as I'll talk about in a second, this other sense called interoception that we don't think about. So they're involved in touch, proprioception, specialized kind of pain, and mechanical itch. But we also were interested in innervation of internal organs. And some of these you're not even conscious of. So for example, you have sensors in your lung that with each breath realize how much your lung is being stretched and control your breathing pattern. Again, you're not even thinking about this because you're not conscious of this lung stretch. You know when things around it get stretched, but not the lung itself. And we know that now without piezo2, this is compromised. And again, individuals without piezo2, especially the very young ones, get hospitalized a lot because they have breathing problems. You also have a neuronal mechanism of controlling blood pressure. So every time your aorta stretches, you have sensors, mechanical sensors there that sense how much this stretch is in effect, and then compensate by regulating your blood pressure accordingly. Again, your body does this without you even being aware of it. And we've shown that piezo 1 and 2 together are required for this process. Now, this third area of interoception is something you are very conscious of. Every time you have to go to the restroom, you feel it. It's because your bladder is being stretched, and these piezo2 activated nerves are firing and telling you to go and relieve yourself. Unfortunately, individuals without piezo2 don't have this sense. And so as they've grown up, they're used to going to they're used to not feeling this and have accidents. So that's why they learn to go to the bathroom about 2, 3 times a day to avoid these accidents. There's so much more to learn about interoception. One of the questions that fascinates me is this concept of fullness. After a large meal, you feel very full. And that sounds like a very mechanical sense, right? But if you look at the published literature, most of the work on satiety, fullness, is all about nutrients, how much protein, sugars, and fats you're eating and sensing. So we know nothing about how much the stomach stretch itself contributes to how much you eat, when you stop eating, how much does this contribute to obesity, diabetes, etc. And so finding these molecules, that sense stretch is going to help us understand new biology, new directions that we did not even anticipate. So you might ask, how does this research help patients? Which is an important question, of course. I hinted at this. First of all, piezo2 deficient patients were completely misdiagnosed to have motor neuron problem. So this research actually helped them identify that they have deficits in proprioception, and there are non-drug related, very simple things you can do to compensate for this. One of them is simply to use your visual system as a replacement for proprioception. And I know some of these patients are benefiting from just this knowledge of what is exactly gone wrong and how they can help themselves. We and others are also developing inhibitors of piezo2, and this could be useful someday in the clinic for chronic pain. And one of the wonderful things about science is it takes you in unanticipated directions, right? So we got interested in mechanosensation because of sense of touch, and now we have programs looking at the role of the same molecules in hypertension, asthma, lower urinary tract dysfunction, etc. So science is fascinating in that way. So just I'm getting close to wrapping up. We think of mechanotransduction mainly from the sensory neuron perspective, but many cell types and tissues experience force, sense, and respond to it. And we know very little about it. My lab and many others have been looking at other physiologies unrelated to nervous system. So we and others, for example, have shown that piezo1, the sister molecule of piezo2 that's required for sensing touch, etc., is important in blood vessel development, in bone density. You might have heard that astronauts who go into outer space actually come back with loss of bone density. And now we know that you need constant pressure on your bones to keep things in normal bone density levels. And piezo1 seems to play a role in sensing this. Many other roles that I won't talk to you about today. So everything I told you about is in humans and mice, mammalian systems. But interestingly, if you look at the evolutionary background of piezo channels, they're present everywhere. They're present in all vertebrates, they're present in flies, in worms, in unicellular organisms even. also in plants. And interestingly, we had one project on looking at piezos in plants. So plants also experience mechanosensation. For example, when roots penetrate the soil and let's say they encounter a hard surface like a rock, they have to sense it and realize if they can go through it or go around it. You might think of this as a passive process just based on physics, but one of my postdocs found that piezo in Arabidopsis, a kind of plant, is actually expressed very highly in the root tips, exactly where this sensing could be happening. And he found out that when he knocks out piezo in plants, the plant's ability to penetrate hard surfaces is compromised. I found this very fascinating that the same molecule that we use for sensing touch is evolved in a very different species in a plant to sense surface hardness by its roots. So evolution is quite amazing. So take-home messages, we identified sensors of temperature and pressure. These ion channels are essential for touch proprioception and pain, but also, as I mentioned before, we found roles in unexpected physiological processes. And it's just gives me an opportunity to remind everyone that, and whole of society should be reminded, is that basic science labs are the nurseries for future medicines. And as an example, I mentioned TRPA1 that was cloned in my lab in 2003, and now it's in phase 2 clinical trials in 2022. So it takes a long time to translate this work into medicines. This is just to put the names of everyone who was involved in this research. As you can see, it takes a lot of people to do research, and it includes my current lab members, all the alumni that have gone on to have jobs in both academia and industry, and collaborators from around the world, and of course, funding. Research is very expensive, and we need we need funding. Anyways, thank you for your attention. Happy to answer, as I said, any questions that you might have.
Again, I will now open up the floor for questions. As usual, we'll prioritize students. The floor is now open.
Thank you for your presentation. I'm Lucy. I had a quick question. You mentioned that mutations cause the absence of PET2. Other than that, do you know of any neurological disorders that might also
Yeah, so I think another types of mutations in piezo2 causes this condition called arthrogryposis, which is a congenital developmental deficit where the tendons don't develop normally. And this is caused by, instead of too little piezo2, too much piezo2. And once again, in the clinic, this was thought to be mainly a problem of joint development. And we found that this phenotype is also being caused by being too much piezo2 or mechanical activity during development, that indirectly from the neuron is communicating with the tendon to cause this phenotype. And this is interesting because, again, the presumption has always been that the deficit is at the tendon level. This kind of opens up a whole new idea of what causes the disease. And although the specific type of arthrogryposis is very rare, overall arthrogryposis is a very large population of syndromes. And again, it kind of opens up question of us asking what else can this cause that has a larger impact. I should also say that autism disorders, often kids who have autism are very sensitive to touch, and they actually respond very negatively. And there's now evidence linking that to touch sensitivity that's also of interest.
Thank you. My name is Robin, and I have a two-part question. First of all, are the piezo receptors different between different species or between different cells within the same individual?
So any gene has different variations in different individuals, but the function of piezos are very well conserved in the sense that they're very tightly controlled. That's why this disease-causing mutation that I talked about, arthrogryposis, it looks overall quite normal, except for its inactivation is a little bit slower. The reason I'm bringing this up, saying that small changes in its function can cause disease. So there isn't that much room for variation of function. It's very tightly controlled. Between species is a very interesting question. We're just starting to address this. And one of the ways we're addressing actually is thinking about what happens to this ion channel when we look at it in fish that live in a very, very deep ocean where the pressure is very, very high. As you can imagine, everything has to be evolved to adapt to that environment. And we're studying this, just started actually, to understand how this, one environment must cause its piezo to work very differently, right? Because its baseline pressure is so different than what we experience. So we're very curious to find how that's changed, and that will actually make us understand how they sense pressure to begin with.
Okay. That's really interesting indeed. Thank you. And that actually kind of brings me to the second part. It's another specific question, but are there any differences between the piezoreceptors, if you know, or if there's any research in this, between the piezoreceptors of the leaves of Mimosa pudica, which is the touch me not plant, which takes its leaves as you touch it, and the root the root ends of the Arabidopsis thaliana?
Yeah. So the Venus flytrap, actually, we studied this too. We have one publication on this. I mean, I presented it in mammals. Piezos are playing the major role in all forms of mechanosensation, but there are other mechanosensors out there. Indeed, I mentioned that how we sense acute pain, hammer hitting your finger, we still don't know. Our lab and many others are studying. It so happens that piezos do not account for the Venus flytrap sensing and closing. We found 3 other candidates that could be, and that work's still in progress to find out which one it is.
Thank you for a great talk. I'm interested in chronic pain, particularly pain due to denervation. Are your sensors involved in denervation pain? What way?
So this is a more of an active area of research. Pain and neuropathic pain are obviously very, very complex. And we actually don't know many of the answers to your questions right now. I think future research will definitely look at this. We are actively looking at this, either from things like UK Biobank that does association studies between genotype and phenotype. So we're starting with the pain phenotype and seeing how this works out. We also can study it in the patients without piezo2, but of course, doing studies on humans very complicated and difficult, and especially if you have only 20 individuals worldwide right now diagnosed, it makes it very, very difficult to do. But in mouse experiments in neuropathic pain models, again, the only thing we know is that any conditions that cause this tactile allodynia doesn't exist in the mutation. That's the extent of what we know right now.
Thank you for such a wonderful talk. It's very, very informative. Thoroughly enjoyed it. I was just wondering if these receptors have been studied in people that have dermographism. You know, you touch them and like they swell up. So are these receptors, has it been looked into? Have a family member, like you can ever so touch them slightly with a feather touch, be it a finger, be it a feather, and you can actually draw on them. Have these receptors been studied, like if they're playing a role and if there is any relation to the immune system, like what is happening? I'm just curious.
Yeah. Not directly studied, but given what we know about piezo2, I could tell you with some confidence that it is what's triggering the output, right? Because this is how you sense touch. But the communication between that initial touch to the response in the immune system, we just don't understand yet. We know that many of these neurons, including the touch neurons and pain neurons, when activated, secrete factors that can, for example, recruit the immune system, because that's how inflammation messages work, right? If you have an injury, your pain neuron secretes pro-inflammatory factors that recruit immune system. So in these individuals, that pathway has gone awry, but we don't understand the connection to touch yet.
Just for curiosity, are these studies you're familiar that are in the pipeline? Curious.
You're getting cut up. I can't hear you very well.
No, sorry. I just wanted to know if you're aware of any studies that are perhaps in the pipeline, or if any of your colleagues looking into it. It's just like an area we know it exists, but haven't been looked into deeply.
Unfortunately, I don't know specifics on that.
Okay, no, thank you so much.
Excellent. So I would like to sort of close off with one final question, touching on something you mentioned earlier about basic science. So one thing that we do here at CCIR is we're really sort of trying to bring students into science and research. And I'm curious sort of what you think of as some of, because you took this to be something that we need to remind society of the importance of basic science. What do you see as some of the challenges that sort of confront basic science, sort of fundamental science, and why this needs to be something that we remind people of and remind society?
Yeah, I think there's a lack of understanding by governments and many non-scientific folk out there of how science works. So I think what the lack of understanding is that every medicine that you hear about, every discovery that has made a huge impact on translational medicine, starts with usually curiosity-driven basic biology question and research. Examples of this are CRISPR. You've all, I'm sure, heard about CRISPR. Has revolutionized how we understand and manipulate the genome, which is starting to become used in various companies, curing sickle cell, for example. And that started by this curiosity of how viruses infect bacteria, right? Has nothing to do with translational work. The people who were working on it didn't even think about it initially. And there's so many examples like this. Another fact is that in pharmaceutical industry, people say that the reason where a drug was initially developed hardly ever goes to the first indication that they try, but many indications afterwards. So I think it's a very known fact that we rely on this curiosity-driven basic science to initiate the process to come up with the next medicines. And yet, I would say, even in the United States, for example, most of funding now, we have to justify that what we're doing has direct application to some drug discovery process. And I just think it's misguided, because we're playing this game where we say this, but it's actually not true, in the sense that what really, really works is to leave scientists some leeway to do the basic science and then see where this goes. And the way I deal with it is every time we have a new discovery and we're being interviewed by either the university or any media, and always the first question is, how will this help, make the next medicine? And I usually answer with the expected path, but I always come back and say, but let me just tell you, this is how science works, and we should support science for science's sake. As I said, discovery from curiosity-driven, how does things work? And then let the applications come from there. So I think it's important to keep emphasizing this point.
Absolutely, and the importance of science education, sort of just understanding how science actually functions, really does sort of fit in also with the theme of today's talks and bring you guys in. With that, if there are no further questions from the audience, I would just like to. Okay, please. So one more question from the audience.
Of course.
So I don't know much about science as a philosophy student, and in Asia, I came across the idea of like quality adjusted life years. The idea that, for example,
Can you come closer to the microphone? You're getting cut up again.
yeah, sorry. So while studying in Asia, I came across the idea of quality adjusted life years. So the idea that someone in pain might have a life that's, for example, like 30% for someone who's healthy. And I was wondering, as someone who often studies the idea of like pain phenotypes or whatever, do you believe that really someone's life can be like that pain could possibly be like quantitatively like measured, and that like someone's life can just be treated like
I'm still having a hard time understanding you. For some reason, the audio keeps getting cut out. I think you're asking about pain measurement and if it could be quantified. So traditionally, people in the clinic use this visual assay for pain. There's like 12 points, and each is associated with a facial expression of how painful it is. And it's really, really not ideal. Unless you've experienced 12 to 1 on the same day, you really can't rank it because experience is very subjective. And we also have the same problem when we're dealing with animal models of how do you assess how they have pain when they have pain. So a lot of people are working on this. There's a lot of work with AI on this, actually. And AI, just by videoing humans' expressions or animals' expressions and looking at many, many data, they actually can come up with a more probably objective assessment of how much pain you feel without even saying it. So it's a very active area of research, and we all realize it's very important to come up with because if you can't quantitatively define it, it becomes very hard to treat it, right? I hope I understood and I answered your question.
Yeah.
With that, I'll do one more final call of questions, just in case we have anything else. If not, I would invite the entire room to give Dr. Ardem.
All right. Thank you all.
Thank you so much. And on behalf of CCIR, I want to thank you again for joining us today.
All right. Bye-bye.